Following GRK2 or GRK3 phosphorylation is the recruitment of arrestins. Arrestins are another group of proteins, so called because they act to block the G protein signaling that happens usually when a mu receptor agonist, such as morphine, binds to the receptor. So arrestins can contribute to this blocking signaling in two ways. The first way is by the first way takes place at the cell surface membrane, and it involves desensitization. So, as we talked about earlier, desensitization is when the receptor, even though being present on the surface and therefore exposed to an agonist molecule, cannot respond. And this generally is because the mu opioid receptor has become decoupled from uh, the G proteins and the G protein signaling apparatus, which is crucial for driving the analgesic responses, perhaps other responses as well. So, firstly, it blocks signaling by contributing to desensitization. It should be noted that GRK2 and GRK3 start this process of desensitization by this introduction of the phosphoryl group. Now, the second way in which arrestins contribute to um, tolerance or contribute to the inability of the mu receptor to respond to an agonist is through internalization. And this is really where the arrestins produce their major effect. So, following the recruitment of arrestins to this phosphorylation site introduced by GRK2 and 3, they can recruit scaffolding proteins which ultimately pull the receptor into the cell within a intracellular membranous structure called an endosome. Now, while the receptor is in the endosome, it can still interact with different pathways and do things, but crucially, it can't react to new agonist molecules, which are on the outside of the cell, which, if you imagine, can reduce someone's reaction to a drug, because if the receptors simply aren't there to introduce the intracellular changes that would usually occur, then you need to have as much of the drug molecules outside as possible to elicit some kind of effect. And you can imagine over time you'll need more and more to try and engage the few receptors that might be left on the surface. So one of the key challenges or avenues explored in mu opioid receptor research is whether we can prevent arrestins from internalizing the receptor. Um, and not only that, whether we can then also prevent the desensitization process, so prevent the decoupling of this receptor from the intracellular G protein signaling. So it's not enough to just have the receptor on the surface. We need to have it on the surface and sensitive to agonist molecules. Um, Arrestin knockouts have shown um, reduced tolerance, and there's definitely room for arrestin targeting drugs as anti-tolerant agents to be used in adjunct with opioids.